All right, we'll get started here in just a second. All right, we'll get started here in just a second. Need that. Okay, I'm going to talk again and see if I fixed it. Oh, the trouble. Okay, I'm going to get started with this stuff. Good morning, everybody. I will ask if there's any questions before I get going with this stuff. We are starting a new unit of material. The test for unit 3 stuff opens. I want to say it opens right at 10 o'clock. If I remember properly, I got it set up so it's right after lecture today. So any questions about any of that? Okay, silence means no. So when I open this up today, it is, instead of just PowerPoint, it's Google Slides, so it's a little bit different. But hopefully I don't know, that's not what I want. We will see. We'll see if this works for us. I don't know. Okay, so this next unit of material, there is a lot of detail. There are a lot of organisms that, you know, some of these guys we have already sort of mentioned a little bit about in passing. We've talked about E. coli and we've talked about Staphylococcus aureus. Those guys, numerous times we've talked about that stuff. And in the lab, it's highly likely that you've thought about and looked at some of those guys as well. This last unit really is, I think, what a lot of people that are enrolled in this course are kind of focusing on in the first place. When we think about the infectious disease, there is a plethora of organisms that set the stage for the likelihood of colonizing and infecting anywhere really within the body is potentially fair game when we think about this. So this last unit of material focuses each chapter on body sites. So we'll think about specific organisms that target these respective sites. And as we progress through multiple chapters, it's highly likely that you will see some of these guys repeated because there are opportunities for numerous body sites to be plagued by some of the same infectious disease agents, oftentimes bacteria, but there are some other potentials as well as we think about other types of microbes like, you know, yeast infection, oftentimes thought of either vaginal yeast infection or oral, so thrush situation, but there is potentials for the Candida albicans to be systemic and target any really organ and cause infection. Typically prognosis when that happens is not so good. So there are numerous opportunities as we think about this to see kind of some of these names repeated. All right. I recommend as we're progressing through here, each of these chapters has some tables with specific organisms and respective symptoms that we might see in these specific chapters from that type of microbe, whatever it is. I pay attention to that stuff. I think a good idea for trying to, get all of these things together is really to start building some note cards and thinking about all of these organisms as we progress through. So I target those tables, and you can see all of that in the PowerPoints that have been posted or in the text. You can see that there are, and in this chapter, I did not put some of the tables that are in the text. I've got the text opened on here, but some of the tables are full of some detail as well as we start thinking about some of this. So initially, as we start with this chapter, we're thinking about skin and eye infections, because really the eye is kind of, we're like, well, where do we put it? It could fit with some of the nervous tissue as we think about that. And there are definitely some aspects of it that we want to think about that way. But it ends up being kind of grouped right into this. So there's a little bit about that as we move through, and some potentials with organisms causing infection. We'll hit some of that. But initially with the skin, and in the last unit, we mentioned part of the innate immunity is the intact skin, providing a barrier to the outside world, keeping some of these organisms in check, not allowing them to take hold and to colonize or to cause infection. And when situations are, unfavorable on our end, or in turn favorable for those microbes, well, the circumstances are changed, and the possibility of some of these guys taking hold and causing issues is apparent. It really kind of depends on what we're thinking about as we move through here. So initially, as we look at this, we've got some acne. This is a common thing that plagues many individuals, oftentimes thinking about, initiation of puberty, starting some of this process as you see excessive oil production, setting the stage really for providing ample nutrients for some of these microbes to in turn thrive. And you may see some of these issues oftentimes tending to clear up after you go through puberty, and in turn there are others that seem to kind of continually be plagued with some issues with acne. And it might be ultimately what you call your own normal flora. What are these organisms living on your skin that sets the stage for some of these situations? It's really pretty impressive. And we'll show here in a second some details, a little bit about some of that stuff that I think is really pretty impressive. So initially, just a quick anatomy of the skin. We showed this, I'm pretty sure, same image before probably chapter 13 when we were thinking those innate defenses. I'm pretty sure we saw the same figure. So we're not going to go through great detail with this, but quickly you can see the layers of the skin, the epidermis, numerous layers of dead cells that provide this initial barrier to the outside world. There are opportunities for sure for organisms to get through their pores, follicles that may lend to entry points. Thinking situations where it's not a wound, not a cut or an abrasion that allows access, just a normal circumstance. And, we could flash back to the issue with the acne as we think some of these pores becoming infected and colonized with some specific microbes that lend to those situations. Here we've got a micrograph that shows the real skin as we move through there. So epidermis in kind of lighter color up at the top, multiple layers of squamous cells all kind of flattened, providing this protection. And from that, moving down into the dermis, so you can see here the really thick layer. And then we're not seeing this, but down below would be the hypodermis, where you see adipose tissue and vessels, so on. So this image is really pretty impressive to look at some of the potential organisms that you might find, and your body, this, thinking the skin initially, as this chapter implies, your skin is an enormous amount of area that microbes potentially can colonize and can call home. And with that said, there are all sorts of diverse areas on your body where some organisms may prefer and others may not. And you move to a different area and the perspective can be completely different when you think about what might be colonizing and what maybe is not. So there's all sorts of different types of organisms that we see here, all kind of color coded out as we move through here. Numerous types of organisms, and we're not going to get bogged down with all of the details. Just think of the tremendous diversity as we look at this. So moving through different areas of the body that have been sampled, and ultimately taking some swabs of these locations, plating this stuff out and seeing what sorts of organisms can we truly cultivate, can we isolate from the body. And like I said, huge diversity from the top of the head to the bottom of your foot and everywhere in between. You can see in these pie charts the representative organisms based off of this key over here. And you can see that the proportions vary dependent upon where we are sampling, all right? And in some circumstances, as we think about this stuff, these organisms, and this is the normal flora, these are the good guys, they provide protection in the fact that they're taking up this surface area, preventing others that might be classified as pathogens from having the capability of taking hold and, moving in to this habitat, if you will. So there are also potentials where some of these organisms on the surface are all fine and good. But if they happen to make their way through the epidermal layers, getting into the deeper layers, to the dermal layers, potentially, where we've got nice rich blood supply, that sets the stage for these guys that might be considered normal flora to now shift over and be thought of as pathogens. So there are all sorts of potentials when we think about that. And as we move through and start thinking about some of these details, that will, I think, become apparent. So more with the acne. So here we can see this handfuls of whiteheads in here. So bacterial infection, there are handfuls of bacteria that may set the stage for some of this. Propionibacterium acnes is definitely a culprit with a species name of acnes. You ought to think acne. It's definitely a common culprit as we look at this, if we were to try to isolate some organism. So it's bacterial infection. You get rash and inflamed hair follicles. folliculitis. I've got a table I'm going to share with you here in just a second that will describe all of these things. So it becomes, can become purulent or superlative, and you've got this formation of this furuncle. So an abscess next door over here is pus-filled lesion, and you can potentially see some of that occurring as well. I think that, yes. So this goes through and talks about the types of lesions, gives us some perspectives here. But before we look at that, let me share this table from the text. Maybe. If I can snag it and move it over. Okay. So this is right out of where it's got to be down at the bottom. Table 21.1. It is not in the PowerPoints, but again, it is right in the text. So it's got some specific terms that we will hit on as we progress through the rest of this chapter. We've already talked about a couple. So the abscess that we just saw, a localized collection of pus. The bulla or bullae are fluid-filled blisters that are relatively small, no more than 5 millimeters in diameter. We've got bigger ones that we'll get here in a second. Carbuncles are deep, pus-filled abscesses, typically the fusion of multiple furuncles. We'll define that here in just a second, but these furuncles fusing together, creating carbuncle. We'll show you an image of that in just a second, and I think you'll be happy just seeing the name and not the image. Crusts are dried fluids from a lesion on the surface of the skin. Cysts, encapsulated sacs filled with fluid, maybe some semi-solid matter or gas, usually just below the upper layers of the skin. Folliculitis, back to those pimples, localized rash as a result of inflammation in those follicles. Furuncles are also called boils. These are pus-filled abscesses, as we see infections, again, in follicles. The macules, these are smooth spots of discoloration on the skin, but smooth is the key. Papules are raised up bumps. Pseudocysts appear as if a cyst, but their boundaries aren't as defined, so more irregularity with that. Something that's purulent means that it's pus producing or suppurative, the ability to make pus. Pustules are the fluid-filled bumps on the skin, so being purulent. Pyoderma is a superlative or pus-producing infection. There are numerous organisms that you might say are pyogenic, so pus-producing, right? Streptococcus pyogenes is a good example of that, and you can see in the back of the throat with strep throat this kind of whitish looking growth, but often, if you were to look deeper, you'd recognize it's a lot of pus that's back there. All right, where did I go? Suppurative, we've already hit that up above. Producing pus, purulent, they go together. Ulcers are before the skin has broken open, an open sore, and you may flip back up to carbuncles with that as well. Vesicles, small fluid-filled lesions, and wheals, swollen, inflamed skin, often itching or burning, maybe as a result of a bug bite. You may see that. So since I've already just got it right in the text, I'm just going to scroll down to these images because that's right where we left off in the PowerPoint with the different types. So the crust, again, the dried fluid on the surface, cysts you can see here, and the epidermal layers as we're looking at that. So all sorts of potentials for what might be in that cyst. Macule discoloration on the skin, but not raised up or anything. The papule, you do have a little bit of a bump, a raised elevation with it compared to the macule. The pustule here you can see the fluid-filled, the pus within that fluid-filled vesicle. The lesion here, this ulcer, and then vesicles and wheals with the respective layers where they are impacted. All right, I'm going to quit with that. Hopefully, now I'll switch back to the PowerPoint and realize I didn't want to quit sharing. All right. Oh, I went right back to the book. That's fine. I should have just talked about the lacrimal ducts right there. All right. Now I'm back to the right thing. Okay, so real quickly, the eye before we start thinking real disease stuff. So we want to hit a little anatomy with it as well to think about some of these potentials in the situation where we may find organisms taking up residence in the first place. Here we could see lacrimal glands providing secretions that in turn, the tears have some antimicrobial properties. They have substances that will make it relatively inhibitory for some of these microbes. There are definitely potentials where they may evade some of these properties and be able to colonize as a result. So there's also the IgA immunoglobulin in some of these secretions that provide some protective mechanisms as well. All right. So with the lacrimal ducts, we've got the movement of the fluid. Here we can see the pores basically that allow this to cycle through with that. And then the nasolacrimal duct that allows drainage as a result. So some microbes may take up residence on the conjunctiva of the eye, as we see this, but the vitreous humor should be sterile. There shouldn't be organisms that we would say should be there in the first place, right? But on the conjunctiva, there are potentials where we may see stuff similarly to organisms that are found on the surface of the skin, kind of the same profile that may be possible colonists here, right? And there's also potentials for infection as well. And everyone, I'm just like, oh, every time I see images of eyeballs like this, it just creeps me out. Conjunctivitis is inflammation of the conjunctiva. We oftentimes think stuff like pink eye. There's a handful of different types of organisms that can stimulate this conjunctivitis. And in some circumstances, pretty contagious. If we're thinking, pink eye, Haemophilus is a good one for that. Blepharitis, inflammation of the eyelids, and maybe you've got a little of both going on together. And then keratitis is inflammation of the cornea, and you can see it looks really pretty occluded here. There's another image, I think it's toward the tail end of this. Now I'll flip the slide and it'll be the very next one, but I feel like it's at the end when we're talking some of the fungal culprits, and there's a stain that they've put in there, and the cornea has picked it all up. It's all, the eye is green. That's pretty crazy. So as we look at this, this is something I hope we've already seen in the lab as we've progressed through here. This first figure is a mannitol salt agar plate, or an MSA plate. These plates are specific really to enhance the growth of staphylococci. It's a type of selective and differential media. So it selects for the Staphylococcus, and it allows us to differentiate between the Staph aureus that will ferment the mannitol and turn the medium yellow, and the Staphylococcus epidermidis over here on the other side. And these guys do not ferment mannitol. They can grow, but they don't ferment the mannitol, and so it stays the same kind of red color that the plate was before it was inoculated. All right. And you can see really kind of a good example with this. The aureus is very good at fermenting that mannitol, and you can see the marker where they've cut the plate in half. Even where the colonies aren't, there is a huge amount of mannitol that's been fermented to the whole plate. If this were incubated a little bit longer, the whole plate likely could turn yellow, all right? Lending to some maybe issues if you don't look at it carefully or quick enough. So that's one super common bacterium that we find on our skin. Both of these, aureus and epidermidis. Aureus also has capabilities of being classified as pathogen. On the surface, all fine and good. Breaking through that and getting in to the tissues sets the stage for staphylococcal infections. And there's all sorts of antibiotic resistance that we see with Staphylococcus. Some of these thinking kind of super bugs, super pathogens, where we've got virtually nothing left in our arsenal of drugs to throw at these guys. They set the stage for some serious concerns. All right. So here's an electron micrograph of the Staphylococci. They often, when we look at them, are clustered up, so bunches of them together, and it has to do with their irregular planes of division, allowing them to be in big clusters like this. Gram stain wise, they'd be purple spherical cells, kind of cluster of grape-like situation. So here's a couple of issues here. Boil, a furuncle, and carbuncle as we move to the next image. Oftentimes, Staphylococcus are the culprit for this, and this would be the Staph aureus, all right? Epidermidis, typically you don't think about that guy being pathogenic in the first place. There are some circumstances where people maybe with an indwelling catheter might get colonized with epidermidis and see some ill effects, but usually otherwise it's just thought of as being normal flora, non-pathogenic, all right? So typically we think of this as Staphylococcus aureus. There's some other potentials as well, but coming in, colonizing within one of these follicles, and then you can see the pus kind of as it's ulcerating out, oozing some of that pus. The carbuncle would be multiple furuncles that have kind of fused together. A lot of this yellowish that we see here is pus, but there's also the staphylococcal organism within that. If you were to take a culture of that, it's highly likely you would find organism within that. So I'm going to move off of that. Maybe I'd rather flip back to that one instead of looking at this. This is a newborn with what we call staphylococcal scalded skin syndrome. So as a result of the organism colonizing and causing serious issues, and part of it, you can see all of the skin kind of sloughing off. We're only looking at the hand here, but I assume the entire body is probably like that. So really pretty disturbing kind of image as we look at that. So this next image is impetigo. Impetigo is a disease that's pretty common, pretty contagious. Oftentimes, young kids, maybe more common to pick some of this stuff up, but it's not truly just them that get plagued with it. But for one reason or another, they are maybe a little more likely to pick up some of this stuff. So the idea with impetigo, there are really a couple of players involved with impetigo. Staphylococcus aureus is definitely a potential. We also think about Streptococcus pyogenes, and oftentimes the two kind of working together to create this perfect situation, this opportunity to cause a pretty good infection. All right. So as we see this, and it's oftentimes on the face where we see impetigo. That is definitely the most common area to see this manifest. So vesicles and pustules, bullae that can rupture, and you see these encrusted sores. Again, really very, very serious situation as we think of being contagious. As far as ultimate ill effects, not so much. It does depend on the types of bacteria that are getting you, if we're thinking antibiotic resistance or not. So I mentioned we saw this, the Staphylococcus in the previous 2 slides back. Streptococcus pyogenes, here's a gram stain of it. And like I said, oftentimes you see these guys working together to establish this impetigo infection, right? So Streptococcus pyogenes, typically we think about these guys causing strep throat. So the fact that they are in the oral cavity and then seeing some of these lesions on the face by the mouth kind of makes sense that it could lend to this. There are potentials that you may have some Streptococcus pyogenes in the back of your throat, and you don't have strep throat. You don't have ill effects. It may be that you had strep throat at some other point earlier in life, and a few of these end up becoming, colonizing and becoming part of your normal flora. You may be classified as a carrier in that situation, for you, although you don't feel infected, you have the potential to spread that infection to someone else and to cause that person to contract strep throat as a result. So it is something that is relatively common to find in your mouth as normal flora, but it also definitely sets the stage for upper respiratory tract infection for sure, and again some other issues like impetigo. We will see some flesh-eating bacteria here in a second, and Streptococcus pyogenes definitely fits into that category as well. So gram stain wise, these guys characteristically are long chains of bacteria linked together, again, gram positive. There are some circumstances, as we look right here, where there's kind of a grouping of them together, right? But as you look around, you do see some individual chains, right? If you really haven't done lots of microbiology, if you haven't looked at lots of gram stains, looking at this and looking at a gram stain of Staph aureus, you might be hard pressed to really tell the difference between the two. If you've got a smear that's really thick, if you've got a lot of bacteria all over the place, it doesn't matter if it's aureus or pyogenes, it's going to look like purple cells all over the place. And it's hard to differentiate. Are these chains or are they clusters? It's pretty stinking sure it looks like clusters because there's so many everywhere, all right? Oftentimes you want to move toward the edge, the fringe, the outer bits of that smear where you can get some stuff kind of spread out a little bit. And if it really is Staphylococcus, you see lots of big clusters of cells. And if you've got pyogenes, you typically see some nice chains like this because they've been thinned out enough that you can see some of these characteristics, right? But there are definitely some things that may confound it. If you look at this, you might be hard pressed to really say what's up, all right? If that's all you're seeing, all right? And in turn, we start thinking about some of the, tests, plating them out on different types of media or some biochemical tests that may help us to differentiate. So you could plate them on MSA, and if it was Staph aureus, it should grow and turn yellow. And if it was Streptococcus pyogenes, it shouldn't grow at all, right? You could also do a catalase test. If you had these guys grown up on a nutrient agar or a blood agar plate, and you've gram stained and you're not quite sure, you could take each colony. Staph would be catalase positive, and the strep would be catalase negative. And you would then go down your specific avenue to truly try to differentiate and see what's up, all right? So there are numerous things that pyogenes can do, all right, besides strep throat, right? And I think deep down, if you had your choice, you'd take strep throat, right? So there are definitely some issues, skin conditions, if that organism can get through that layer and, enter a cut or a wound. Cellulitis is this first one that we've got here, painful red rash. There are numerous bacteria and other things that can stimulate cellulitis. It doesn't have to be really microbial, but oftentimes it is, and pyogenes is definitely one of the culprits. Ehrlichia, raised rash, clear borders. Here we've got a pretty good indication of that. And then erythema nodosum, red bumps and nodules, typically lower legs. They are different than cellulitis. So those are some potentials. This is the bad one. So this is necrotizing fasciitis or flesh-eating bacteria. Streptococcus pyogenes is definitely a culprit for this. There are other bacteria that are also possible to get in and cause some serious issues, right? So the first image is the infection before we've gone in and debrided. That's what we're looking at over here. So there's all sorts of nasty infection as we look at this. A lot of the dark brown, blackish lesions here are necrotic tissue. This is dead tissue that needs to be removed. And if not, it's going to lend to more serious potential fatal consequences as a result. So here, basically debriding, it's the same patient where they've gone in and removed or are starting to remove all of the infected tissue. And yeah, really kind of disturbing to look at that. So getting into the fascia is basically where this organism gets in and causes a serious issue. The deep tissues basically prime ground for this organism to have access and take over, if you will. So there's definitely potentials with this for amputation, if not, able to be dealt with. And there are numerous occurrences where we see someone with flesh-eating bacteria and they don't recover, all right? The infection is too great. And we think, potentials with these organisms to wind up with septicemia, where the bacteria gets into the circulatory system and courses throughout the body, sets the stage definitely for fatal consequences as a result. This next one, both of these images are the results of a bacterium called Pseudomonas aeruginosa. This is a gram-negative rod. Somebody you've probably seen a little bit in the lab, I think. So there's numerous potentials with Pseudomonas. It's an organism that is relatively common in the environment. It likes wet environments. It likes moisture. It likes warm environments. It likes hot tubs. And I think, maybe a hot tub that's poorly maintained sets the stage for some of these issues. So I think if it's adequately chlorinated and dealt with, the potential for it is minimized for sure. So this is called hot tub folliculitis, basically because these bacteria tend to thrive in this type of an environment. So sores, little raised bumps that are, look pimple-like as a result of these guys, all right? And, as a result of, taking a dip in the hot tub a few days prior. The next image, same organism, Pseudomonas aeruginosa, is also a pretty good organism to cause ear infections. Depends kind of where, but Otis externa. So the outer ear. Swimmer's ear is a common name for this, and Pseudomonas is one of the culprits, pretty common culprit for that. There are some others that also could be potentials. Pseudomonas, as we think about the ear infection, sets the stage for some issues because it just normally is pretty drug resistant. There are only a few antibiotics that are effective at targeting Pseudomonas in the first place. That's an issue. It also produces a very good capsule layer, so a sugar coating. It participates in biofilm formation and basically shrouds itself under these sugar coatings that make it almost like a shield or a barrier to those antibiotics. It kind of encases them and protects them so that the treatment is ineffective, really, when we think about that. So that's potential. So could you get both from soaking in the hot tub? You better believe it. Definitely an issue. So here we've got some images of this Pseudomonas here growing on a type of medium called XLD, xylose lysine, sodium deoxycholate. Nice, shiny, it's kind of shiny looking up on the big screen. This characteristic when we look at these colonies, seeing sort of the shiny or the glistening colonies is very indicative of a capsule forming bacterium, a sugar producing organism that's got, again, kind of a leg up at avoiding our immune response because it can, well, it can form biofilms and it can go undetected by some of those phagocytic cells. All they see is sugar, right? Here's another image, and it depends on the type of medium. Obviously, in this XLD agar, you're not going to see it, but on something like a nutrient agar, it's pretty common to see it on nutrient agar. As Pseudomonas grows, it can produce a pigment called pyocyanin. This pigment is kind of blue-green, and it is indicative of Pseudomonas, all right? Seeing this type of pigment produced on that medium is a pretty good indication that's what you got going on, all right? It also has some kind of characteristic odors, not that you should be going around smelling bacterial cultures, but Pseudomonas definitely has a smell that's a little bit different, all right? Most bacteria smell terrible, right? They do not have an odor that's not off-putting. But Pseudomonas is a little bit different, right? And there are potentials that some say it smells like grapes, grape chapstick. Others say tortillas, corn tortillas specifically. That's what I smell, is that. It's interesting, all right? And most of the other types of bacteria, they don't smell like that. So, interesting. They also may, under specific circumstances, like a black light, can produce fluorescent, which will fluoresce under UV light, proper conditions, right? So definitely a potential to maybe key you into some of that. I was teaching a pathogenic microbiology lab, so we were dealing with lots of disease stuff. After your intro lab, where you're introduced to all of these things, the pathogenic lab focuses more on isolating and cultivating all of these pathogens that we think about in this last unit of material. And I had a student that had a flashlight on his keychain, but apparently this flashlight had multiple things on it. You could switch it up, one of them being a black light. And he would, I'd have a rack of bacteria up there, handfuls of different unknowns that we would be working on, and he'd pull his flashlight out and shine it. And one of the test tubes would glow, and he would take that one. Why? because he already knew what the hell he had. He had Pseudomonas, and it was a done deal. At first, I didn't realize it at first, and then I was like, wait a minute, that's not fair. You can't do that. But it worked pretty good. All right. All right. Let's stop here. Let's stop. Let's quit with pimples. That's a good stopping point. Any questions or concerns? Okay, guys, stop that. Have a good weekend. It's finally getting nice out there.